Good morning. I will start my talk introducing the consortium working to this Human Frontier Science program project. So the project was funded started in December 2021, and it will end in the next month. And this project, so I will present you not just my results, but the results of all the consortium. So this consortium is coordinated by Professor Henry Hess from Columbia University. Harry is a mechanical engineer that uses mechanical engineer principle to model the behavior of macromolecules. And that's me. I come from the University of Pisa. I have a special expertise on neuromechanotransduction. I'm a molecular biology. And then there is Horit Sheffi from Baral University. She's a neuro engineer, expert of neuroregeneration. And then there is Akira Kakugo from the Tokyo University, and is a special expertise in modeling the dynamics of microtubules in vitro. So why we decide to be partners in science? Of course, we are driven by curiosity, and the subject of our curiosity, the subject of our curiosity are neurons. So neurons are mechanosensitive cells. They react to mechanical stimuli. And what we know is that neurons in our body during the whole duration of our life are constantly subjected to mechanical stimuli, to cyclic and repetitive mechanical motion. And these mechanical stimuli cause damage to neurons. So if we think to mechanical machines, for example, our washing machine, you know that the washing machine works for many cycles. At some point it breaks, we throw away, and we buy a new one, or we can replace the piece and continue to use it, okay? So what happens to the living organism? Of course, we are different from mechanical machine. We cannot be throw away when we break, hopefully. And the point is that we couldn't replace our neurons if they fail. So this means that nature has been evolved to give us molecular mechanism, to avoid loss of neurons. So neurons should have mechanism to react, to counteract these mechanical stimuli. So we have a special focus on microtubules. Why microtubules? Because microtubules are the rigid element within the neurons. They are most subjected to these mechanical injuries. And so we want to really know how our neurons can survive for all the life, okay? So moved by our question, by this question, we came together to try to elucidate this mechanical mechanism, this molecular mechanism. We started with doing some simulation study to predict the mechanical behavior of these microtubules under cyclic repetitive mechanical motion. And going back to mechanical machine, for a machine, for example, piston engine or washing machine. So we can plot this fatigue plot, which can predict us the probability that the machine break under a number of cycles, okay? So you can do the same also with a molecular machine. For example, here you see a myosin motor, and you can build this plot and experimentally evaluate that this motor fail after 10 to 6 cycles. So the point of Harry was, can do the same with the microtubules. Of course, for doing this, you need to understand which is the structures of microtubules. So you may know that microtubules are composed by alpha and beta tubulin monomers, and is organized in 13 protofilaments to form the microtubule structures. Okay, within these structures, so within monomers and within protofilament, there are bonds, okay? And these bonds break and create continuously, okay? And there is the theory, the Bell equation theory, that can allow us to predict the half-life of these bonds And based on this notion, we can predict the fatigue, and we can plot this fatigue behavior, okay? Of course, this is a model. The model needs to be experimentally validated. And this brings to me to explain you the work made by another partner at Kakugo Lab. And so you see here Kakugo and Rubaya, the people working on that. So what they did is to start to address this problem by studying microtubules in vitro. Okay, so what they did is to grow microtubules in vitro on this flexible PDMS substrate. And what they did is to make a cycle of compression and relax, compression and relax. This repetitive cycle of compression induce effect on microtubules. You can see that after some cycle, what happens during compression is this effect buckling. You see the microtubules form some wave, and when they relax, at some point they break. You can see the arrow. The arrow point the break points. Well, by measuring this breaking behavior, you can build this experimentally, you can build this fatigue plot, exactly as I showed before. And what came out is that this plot depends, of course, on the, gives the probability of breaking as a function, the number of cycles, but it depends on the strain. What means the strain? The strain is the amount of compression. So you can compress of 5%, 10%, 50%, so depends on the strain intensity. What we also know that when we expose microtubules to this mechanical compression, what changes is the interaction between microtubules and microtubules binding protein. And for example, what you can see here are motor protein, kinesin and dynein. And you can clearly see that after a compression, depending on the strain, the amount of the strain, what changes the velocity of these motor proteins. So this should have an impact on living cells. Well, the next steps, and this I came to my job, is to test what happens with the living neurons. For doing this, we need to have a machine for inducing this repetitive mechanical compression. And this machine was generated by Kakugo sensei, and this is a microstretcher. What you can see here, there is this microstretcher within a stretching device, which is equipped with a micrometric screw for precising control the strain amount, 5%, 10%, so we can get control on this parameter. Well, this device was, within the project, this device was first optimized in vitro. So what we can see here is the device. On the top of the microstretcher, we put PDMS in a pre-stretched PDMS. Then we can grow microtubules on this surface, and we can apply the repetitive cycle at different strain. And what we can observe that as consequence of this compression, microtubules buckle. You can see here the waves. And what Kakugo and Rubaya did is to measure the amplitude and the wavelength of this buckling, okay? And what they found was that increasing the strain, what happens, there is an increase in the amplitude and a decrease in the waves. That is much expected. An interesting point is that what they did is to use microtubule binding protein, because in presence of MAPS, what happens is the microtubules reticulate. So you don't have any more single microtubules, but you have bundles. That's what you see in neurons, especially in the axon. Okay, and what they observed was that. When you have, for example, tau, but not MAP4, the mechanical fatigue, the mechanical behavior of these microtubules change. They are more resilient, more resistant to the mechanical stimulation. And this is pretty interesting, thinking to the living neurons. So the next steps was really to perform the experiments on living neurons. And these experiments were performed by Allegra. She's in the audience. Please visit her poster. She will tell you much more about her project. And what we did here is to, as you can see in this picture, we used pre-stretched PDMS onto the macrostretcher. Then we put onto the top axona a silicone chamber because we want to create a microfluidic chamber. And in one chamber of this microfluidic compartment, we see neurons that are dorsal root ganglion neurons. And what happens is that neurons can grow through the channels. Okay, these channels are big enough for allowing neuron axons to go inside, but the cells couldn't pass through them. So you have the channels with a pure preparation of axon bundles, and you have the axonal compartment where you can see isolated axons. Well, what we did here was to make a cyclic compression. So the PDMS was cyclic, moved from the pre-stretched position, rest position, pre-stretched, resting position. As for the neurons, it was the opposite. They passed from the rest position to the compressed one. Rest, compressed for many cycles. How many cycles? We tested 20 cycles, okay? But we tested different strain, 2%, 5%, and 10%. Why 2.5%? Because in the literature is predicted that this strain is well tolerated and the neurons could survive. And so we tested 2.5%, then 10% as a highest strain, and 5% as intermediate strain. So let's show some results. I will start with the 10%, which is the highest stimulation. I don't know if you are familiar with transmission electron microscopy, but who is familiar with that can clearly appreciate that these cells are totally destroyed. So if you look to the nuclei, you can see that the nuclei are totally condensed. There are really degeneration in nuclei or organelles. You lose the membrane, you break everything. And also, if you focus in the axonal compartment, you clearly see that you lose the ultrastructure that you can see in control axons. So we can conclude that this stimulation is destructive. There is no way for the neurons to recover from this damage. So let's see what happened with the 2.5%, which is the lowest strain. So looking in the ultrastructures, axons seems perfectly healthy. Okay, so there is no damage in the structures. Okay, however, we see a reduction in the length. Okay, so something happens. There is a retraction of the axons, and this when we focus on the stability of the microtubules, we can follow the stability of the microtubules by following the post-translational modification. And so what we saw is that there is an increase of stability. So what happens is the axons increase the stability of the microtubules to react to this decrease in length in order to recovery the length. In fact, this is going to happen 24 hours after the stimulation. Okay. As for the intermediate damage, if you look to the 5%, if we look to the ultrastructures, in this case, we see damage. We see that microtubules twist, we lose microtubules, the microtubules interdistance increase, there is a disconnection of axons, so it is a severe damage. And also, if we look to the stability, in this case, we see that there is a decrease in microtubule stability. So this is a very severe alteration. So the question was, can the neurons recover from this damage? And we reanalyze all this data 24 hours after the damage, and incredibly we saw that they go back to the initial situation. So in terms of stability, ultrastructure, everything is recovery. So the question is, how is the molecular mechanism? How they can do recovery from that damage? And we perform some RNA-seq analysis, gene ontology enrichment analysis. What we found that there is one pathway, the RAS pathway, that is dysregulated. For making a long story short, what happens is that soon after the damage, the axon retract, they lose the adhesion point, and this causes an activation of RAS. But what happens is that the cells overexpress GEF acting on RAS. GEF are guanine exchange factor that are responsible for the transition from inactive to active form. And because of that, in 24 hours, we can recover the length and we can recover the stability of microtubules. So we came out with our model of neuronal resilience. So this is the model that we propose at the end of the project. So in physiological condition, when we have mild injury, mild mechanical stimulation, what we see is there is axon retraction. This trigger increasing stability of microtubules, and we know that the stabilization of microtubules is the most powerful mechanism to promote axon elongation, axon growth, to recover the initial situation. With mild injury, what happens is that there is the retraction, there are damage to the microtubule structure, RAS inactivation, the cells respond by overexpressing GEF acting on RAS, and this trigger axon elongation with well-known mechanism. Unfortunately, when the mechanical compression is too high, this happens, for example, during injuries, there is no way to recover. Well, I finished my time. I skip this just to tell you that there is another part of our colleagues that are working also on making dynamic study on living microtubules. This is still ongoing, and I will come to the end with knowledge people. If I have just really 10 seconds, I want to tell that this project is ended, but a new project is starting, and so I'm rolling 4 postdoc positions in my lab. And if you know people that may be interested in these keywords, please feel free to share this news. Thank you.